Hi, this video is intended to give you enough background to successfully do the experiment on diffusion and osmosis. Notice that in this video we're going to just be looking at some of the generalities of the experiment, and you'll need to follow along in your lab manual for a lot of the details. So first of all, what are diffusion and osmosis? Well, diffusion, and we'll start with this because it's a little simpler, is basically the movement of solutes, so things dissolved in a solvent, from regions of high concentration to regions where they're less concentrated. Now you might see this if you put a drop of dye into a beaker or a glass of water. What happens is you'll see that dye be very concentrated at the point where it hits the water, and then it spreads out from there. And over time it will spread out more and more until eventually it's completely spread out evenly throughout the glass or flask or whatever. Now, the reason it does this is because the molecules in the water are moving around randomly and bumping into each other, and the effect of this random, what's called Brownian motion, is that the molecules, although they're not moving in any coordinated fashion, eventually the dye molecules start out all concentrated together, and they eventually spread out. And this is just a result of this random motion. Diffusion can also happen across a membrane, where we have highly concentrated type of molecule on one side of the membrane and a low concentration of that molecule on the other side of the membrane. And the molecules will move back and forth through the membrane, but the net effect is that the molecules that are in the more highly concentrated region will move preferentially into the region of low concentration. And some of the molecules will move in the other direction, but the net effect will be to equilibrate those two regions. Now let's look at osmosis briefly. Osmosis is different from diffusion because remember that osmosis was the movement of the solute, the things dissolved in the water or solvent. And osmosis is the movement of the solvent itself from regions of high solvent concentration to regions of low solvent concentration. Okay? Now, this often seems a little weird to people because you're not used to thinking about the concentration of the solvent itself. You're used to thinking about the solvent. But in biological systems, this is really important, and in biological systems, this is almost always water. Now, imagine that we have a partially permeable membrane, as you can see here, and on one side, on the left side, we've got a dilute solution of, say, water, or maybe it's even pure water, and on the right, we've got a solution of something else like salt or sugar dissolved in the water. Now on the left, we've got a high concentration of water molecules, and on the right, we've got a low concentration of water molecules because many of the molecules form a hydration shell around the solute molecules. So, as with diffusion, the molecules that can move, in this case only water, can move across the membrane from regions of where they're highly concentrated to the region where they're less concentrated. And so therefore they move preferentially, and the net movement is from left to right in this diagram. Now, in a cell, this happens because the cell membrane acts as that partially permeable membrane, and it controls what goes into or out of the cell. In the case of a plant cell also has a rigid cell wall, but that cell wall doesn't really control what goes in and out. It's quite permeable and things can move through the cell wall. But plant cells also have a cell membrane, and that cell membrane is inside the cell wall, and it does tightly control what goes in and out. Now, water is one of those things that can move across a membrane. And so in the case where you have a solution of say just plain tap water, then water is going to preferentially move from outside of the cell where the water is really concentrated to the region inside the cell where the water is less concentrated. And as a result, the cell membrane will push against the cell wall, and it will result in the cell swelling and becoming turgid. Now, in an animal cell, this can often even lead to rupturing the cell membrane, because the cell membrane doesn't have much support, doesn't have nearly as much support to it as a plant cell does. It depends on the cell type. If you take a plant cell and you put it in what we call a hypertonic solution, that is a solution with a lot of solute, then the net movement of water is from inside the cell to outside the cell. Remember, some does go from outside in, but more comes from inside out. And as a result, the contents of the cell shrink, and all of the chloroplasts and all of the other cell contents get essentially squeezed into a smaller and smaller space with the cell membrane surrounding them. And so we'll actually, in another lab, we'll actually look at this under the microscope and see the difference between a turgid cell and a plasmolyzed cell, such as one that's been placed in salt water. Okay, so let's get into what we're actually going to be doing in the lab this week. This lab is really simple, but it takes a little bit of time, and so when you arrive in class, I want you to get going as quickly as you can with it. And there are basically two experiments. The first experiment looks at diffusion through dialysis tubing. Okay, so dialysis tubing is a semi-permeable membrane that is used for dialysis patients. So people that have lost their kidney function, you can basically filter out the toxins and metabolic wastes that accumulate in their body using dialysis tubing. And so what we're going to do is we're going to fill a beaker with about half full with water, and we're going to put some Lugol's iodine solution in there. Okay, remember that the Lugol's iodine solution is a test for starches. Then we're going to take some dialysis tubing and we're going to seal it at both ends and put into it a mixture of water, starch, and glucose. Okay. Following that, we're going to just put make sure it's well sealed at both ends, and we're going to put the dialysis tubing into the beaker with the iodine and water, and we're going to let it sit there for about an hour. During that time, some of the substances that are in the beaker or in the dialysis tubing might be able to move through the membrane. Okay, and so you should be able to predict which substances are able to move through. After an hour, we're going to do a test, as to whether there is starch or glucose in the beaker or in the dialysis tubing. Okay, so you've learned in previous weeks how to do the test for starch, and you've also learned how to do a test for glucose. So we're going to do both of those to determine which ones of them passed through the membrane. In the second experiment, we're going to be looking at osmosis into and out of potato cells. And this is, again, another very simple experiment. And to do this, we're going to cut little cores of potato and weigh each one of them. Okay? And it's going to be really important that you're careful about weighing them accurately and then not mixing them up. Once you've weighed them, you can place each of the core in a different solution of sodium chloride. And the sodium chloride concentrations will be 0% sodium chloride. This will be just plain water. 1% sodium chloride, that means basically one gram of sodium chloride per 100 grams of total solution. And we're going to do it for these other solutions. We'll leave them for 60 minutes. This will allow the water to move into or out of the potato cells, as is going to happen in the various beakers. And then after about an hour, we'll take them out and we'll reweigh them. And we'll be able to see which potato cores gained weight and which ones lost weight. Okay, and for this experiment, we're actually going to graph our results. And what we might see is something like this, where we have some potatoes in a low concentration of sodium chloride, and they may, in this case, they gained weight. Okay, and then others in a highly concentrated sodium chloride solution lost weight. Okay, now one of the interesting things about this is that we can draw a regression line through these data points, and I want you to think about what the point where the regression line crosses the axis. What does that point mean? Okay, so think about what that point means, because you're going to analyze your own data, and I want you to be able to come up with an answer to that simple question. Okay, I will see you in lab.